ahead.
Good morning, everyone. My name is Ben Domingue. I'm an assistant professor at the Stanford Graduate School of Education. As was just indicated, I'm going to be giving a talk about an analysis of genetic similarity amongst friends and schoolmates in the National Longitudinal Study of Adolescent to Adult Health. If you have any questions during the talk, feel free to interrupt me. So this project is motivated by the idea that we observe genetic stratification across human relationships. So we have evidence that genotypes cluster into both friendships and spouseships, and this evidence comes from a variety of different genetic analyses. We have evidence from candidate genome studies, whole genome studies, and studies looking at trait-associated loci. One reason you might worry about this is that such stratification could have implications for gene discovery and disease prevalence, and a few people have indeed looked at that. I'm going to look at something a little bit different. I'm going to ask why we observe this genetic stratification and ask about its implications for GWAS interpretation. This work is done in the context of the AdHealth study. This is a longitudinal study in the US. It began in 1994-95 when a number of schools were sampled for inclusion in the study. Within each school, in total, roughly 20,000 students were chosen to be followed longitudinally. We last interviewed these students in 2008. Wave 5 data collection is actually going on right now. During that last data collection, we attained biospecimens with which we could produce genome-wide data. We also observed what are nearly complete educational attainment records, and height and weight were measured, and with those two we can obviously derive BMI. So I want to focus in on two key design features of the AdHealth study. The first is the friendship data. So AdHealth is a school-based sample. There were over 100 schools in the sample. And while over time we only follow a subset of all respondents, at the first point of observation, they talk to everybody in every school. And so a respondent at that point can nominate up to 5 female and 5 male friends. And with that data, we can build rich social networks of the friendships that exist within each school. And I've given you kind of an exemplar of what that might look like at the right. One reason this is really convenient is that we can actually observe non-friends with this design. In general, studies about genetic stratification across human relationships have been limited because we don't observe the people you're typically not married to, right? We observe your spouse as a function of the survey design because they're typically household surveys, but we don't really have any information about the other social people in your world that you did not marry. So this is a real benefit of working with the AdHealth data. The second design feature that I've already kind of referenced is the fact that we have genetic data. This was derived from saliva collected in 2008. Respondents were genotyped using 2 chips for historical reasons that I won't go into. We have roughly 10,000 genotypes, and the analysis that I'm going to describe is based on roughly 600,000 SNPs common to those 2 chips. I'm going to focus primarily on the sample in that bottom right circle. This is roughly 3,000 unrelated European people who are in roughly 4,850 friendship pairs. Because of what we're doing, we need genetic data on each of the friends, and given the nature of the random sample, we observe way more friendships than we do friendships with 2 genotyped respondents. So this is a greatly reduced subsample of the full social network data, but it's sufficient for our purposes. So I'm going to use polygenic scores. I imagine most of you are familiar with these. Very briefly, what I'm going to do is just count the number of trait-associated alleles that you have across the entire genome and use a weighted average of these to create a single number index of your genetic predisposition towards some outcome. I'm going to focus on polygenic scores for 3 outcomes: educational attainment, BMI, and height. The GWAS that I used to construct these polygenic scores were all done very recently using very large samples. Notably, AdHealth was not used in any of these GWASs. In constructing the polygenic score, I'm just going to use all genotype SNPs. I'm not going to use any imputed SNPs. I'm also not going to use, I'm not going to remove any markers for LD considerations or because of p-value considerations. I'm just going to do a very simple thing where I use all of the markers that are available and then weight using the GWAS beta weights. After constructing polygenic scores, I then residualize on the top 10 principal components and standardize the polygenic scores to have a mean 0 and standard deviation 1. The scores all predict outcomes very similarly in this data. So The three scores each are correlated with their respective outcomes at around 0.25. Okay, so the key feature of what I'm going to do here is I'm going to use two measures of the social genome. The first measure is going to be something that tells me about your school. In fact, what I'm going to do is I'm going to construct the mean polygenic score of all of your schoolmates, with the one caveat that I don't have all of them in my genetic sample, but I'm going to construct the polygenic score for all of the schoolmates that you have in the genetic sample. The second measure of the social genome is going to be based on just your friends. So just focusing on those people with whom you have a friendship, I'm going to construct their mean polygenic score.
Sorry, I missed probably the first part of the presentation, but how you define friendship?
That's a good question. So at the first point of observation, everybody in the study could nominate up to 5 male and 5 female friendships. What we did is basically identified friend pairs as those two, any two people that had any declared friendship link between them. So if I said somebody was my friend, or if that person said I was their friend, we're counting that as a friendship for what I'm going to describe. In a sensitivity analysis, we also look at only those friendship pairs where both people nominate the other party, and the results are largely similar. And so since using the slightly weaker definition of friendship allows us to use more data, we have privileged that in the write-up, but our results seem robust
And they're both male and female, or only male friends?
No, they're mixed sex friends.
Mixed, okay.
You were limited to you were limited to 10 nominations in total. 5 of those could be female and 5 male. But that's not really a feature that I mean, this sex thing was just a function of how they defined things back in the 90s when they collected the data.
Okay, thank you.
Mm-hmm. Okay, so the first question we address is whether friends are genetically similar, and we do in fact observe friend-level associations for BMI and educational attainment scores. So the educational attainment polygenic score of the respondents in our sample was correlated with the mean friend polygenic score at around 0.06. You can also see a slightly reduced correlation between the BMI polygenic scores of our respondents and their friends. Moreover, you observe correlations between individuals' polygenic scores, especially for educational attainment, and that of their friend. So an individual's polygenic score in our sample is correlated with their schoolmates at around 0.14. Now, what you don't have here is any accounting for social structure. We know that people aren't randomly assigned to schools, and so we need to probe what the role of social structure is here a little. So the question I want to ask is how much of the observed genetic similarity between friends is due to the fact that schoolmates might be genetically similar? So I'm going to get at this via two fairly simple models. The first model involves prediction of an individual's polygenic score based on the polygenic score of their friends. That's that bit in red. I'm then going to also consider a second model, M2, where I now add the mean polygenic score for the schoolmates. That's the bit in blue. And the goal is going to be to compare estimates of the B, the red part, between M1 and M2. In particular, I'm going to expect attenuation as I go from M1 to M2 if there is, if school concentration of genotypes is actually an important part of the story. So what we observe is that a substantial amount of the friend similarity that we get is due to school clustering. So if you look at the dark bar for each of these three panels, that is the estimated effect of your friend's polygenic score on your own. And this is only significant for educational attainment, so let's focus there.
Dennis, can you speak a little bit up? So somehow you are now a little bit.
Oh yeah. Let me. I'll try to get closer.
Yes, but no, it's better. No, it's better. Perfect. Thank you.
No problem. So what we see for educational attainment is that while there is some predictive power of your friend's polygenic score on your own, that power is greatly diminished once we, once we control for the genetic environment of the school at large, right? So it seems to be the case that the assignment to school is very important in our understanding of the degree to which friends are genetically similar. We can also observe this when we consider genome-wide similarity. This is something I discuss more in the paper than I'm going to talk about here, but I want to at least note it. So we observe that friends are more genetically similar than just people chosen at random, but it's also the case that schoolmates are just more genetically similar than people chosen at random, and that once we account for the fact that schoolmates are more genetically similar than random people, we observe a much less genetic similarity between friends. So the punchline is that social forces are doing a fair bit of work here. It's not the case that we have a story that's very consistent with active selection by individuals into friendships with people that are genetically similar to them. What we see is a situation in which society is, forcing people into environments with already genetically similar people with whom they then form friendships. Okay, so thus far, the story I've been telling is about genes and environments. So we observe some gene-environment correlation since friends resemble each other along key genetic dimensions. Moreover, schoolmates resemble each other. And this seems due to larger structural forces. I'm now going to start talking about genes as environments. So there are reasons to suspect that the genes of one's social alters also matter for an individual's expressed phenotype. These are called social genetic effects.
Sorry, can I ask a question about the slide before? And so, do you have any negative control to claim that these genetic similarity are specific to educational attainment rather than being a similarity in the overall genetic profile due, for example, of similar population structure, similar ethnicity?
Yeah, that's a good question. Well, okay, so first off, we don't really observe genetic similarity so much between the height and BMI polygenic scores amongst friends. So I think that's one kind of important part. There, we observe a stronger genetic similarity on the educational attainment polygenic score than the other anthropometric scores. So that's one component. When I'm talking about the overall genetic similarity, there's certainly a role that population stratification is playing here. The measures that I'm showing in this figure, these are based on King relatedness estimates that are somewhat robust to population stratification, but I do think that certainly plays a role in what I'm describing. And so I think that, parsing out exactly how much of this is due to population stratification is challenging. I consider several different measures of genetic similarity in the paper and computed across a few cuts of our data, and we get largely similar stories that the genetic similarity of friends seems driven mostly by the genetic similarity of schoolmates, but I'll acknowledge that the population stratification is tough. You'll see a little bit more of the negative control part in what's to come, I think.
Okay.
Okay, so as I was saying, what I'm going to talk about now is basically the idea that genes can effectively function as environments, so so-called social genetic effects. I became interested in this based on a recent mouse paper that you all may have read. So it turns out that a mouse's phenotypic expression depends on the social genome defined by its cage mates. So if I have two mice, each of them is going to experience some influence from its own genes. But if I put those mice together in a cage, it could be the case that the genes of one mice also affect the outcomes of the other mouse. In fact, in that paper, they showed that you overestimate the heritability of certain traits if you don't account for these social genetic effects, right? So this is the jumping off point. That paper is very clean because what you can do is you can exogenously assign mouse to cage, but my IRB will not allow me to exogenously assign humans to friendships, schools, or certainly marital relationships. Right, so identifying causality when we have strictly observational data is much harder. One way of seeing this is to think about the idea of the social contagion of obesity. This is something that came to prominence a number of years ago due to an article in the New England Journal of Medicine. One of my co-authors on this paper, Jason Fletcher, wrote a follow-up basically showing that using the same methods, you can detect social contagion of a number of things that seem a little less likely to be socially contagious. And we're going to use a kind of similar cross-trait analysis here. So what we're going to do is consider a number of traits that have a kind of range of a priori hypotheses about the extent to which they should be affected by social genetic effects. And we're going to use that cross-trait analysis to probe the degree to which social genetic effects may occur. So the question is going to be whether social genome predicts individual phenotypes net of own genome, right? And we're going to get at this via two very simple models. In both of these models, we're predicting one's own phenotype based on their own genotype. That's the B times GI, plus something about the social genome. In model 3, we consider the social genome as it pertains to friends. In model 4, we consider the social genome as it pertains to schools. So here's what we find. Let me walk you through this figure. The dark bar in each of these two panels is the predictive power of an individual's own genotype on the standardized years of educational attainment. The lighter bar is the predictive power of the social genome. On the bottom, it's the mean polygenic score of your friends. On the top, it's the mean polygenic score of your school. Right? And so you can see that we get a fair bit of predictive power from the social genome. In fact, in the raw metrics, I think it's pretty marked. We find that a 1 standard deviation increase in your own polygenic score predicts an extra half year of schooling. In contrast, a 1 standard deviation increase in your friend's polygenic score predicts an extra third of a year of schooling net of your own polygenic score, whereas a 1 standard deviation increase in the school polygenic score predicts, again, almost half a year of educational attainment net of your own polygenic score. There's also some evidence of attenuation in the estimated effect of your own polygenic score. So you can see this via that dashed red line. The dashed red line is the effect of the individual level polygenic score on the individual level phenotype in a model with no other controls. And what you see is some retraction from that line when we include controls for the social genome. In contrast, when we consider BMI and height, we see really no evidence for social genetic effects. We see, the estimates of the social genome are all pretty much zero once we account for uncertainty, and there's really no kind of attenuation in the estimated effect of your own polygenic score. So basically, to sum it up, the social genome as measured by educational attainment polygenic score predicts own educational attainment, whereas the social genome as measured by height or BMI polygenic scores are irrelevant for one's own phenotype, which is, I think, fairly consistent with what we would expect a priori.
Sorry, can you repeat how you construct the social genome? It's not very clear to me.
So using the social networks that we have, so the first thing I do is I construct the mean polygenic score for every schoolmate that you have that's in our genetic data set. For the mean polygenic score of your friends, I just take those people that are in, that have some observed friendship tied to the individual and take their mean polygenic score. Does that make sense?
Yeah. Okay.
So in the paper, we describe a variety of robustness analyses. I only want to touch on a few of these because our story is largely consistent across all of them. So one thing we examined is whether or not these findings are true within school. And so we did some within school analyses, and what we observe is that when we only examine within school, we still see that the effect of your friend's polygenic score remains predictive of your own of one's own phenotype, right? So there is something very specific about friends. We also looked at whether or not this was a story that was perhaps driven by overall genetic similarity and not the educational attainment polygenic score. And in fact, there does seem to be something very specific about the educational attainment polygenic score. So when we use some GCTA and Grimmel analyses, we get similar results that suggest overall genetic similarity is not driving our results. It really is the educational attainment score. We finally considered results in increasingly homogeneous samples. So I based on PCs, I just took an ever kind of more homogeneous group, the kind of increasingly hot colors in that figure. And when we reran the analyses based on data just in those, those kind of Russian doll-like eggs, we get very similar findings, right? So if you look at the coefficients in this supplementary table, you will observe that in fact we don't see huge changes. Things are statistically significant everywhere. The story is quite robust to taking more homogeneous samples. Okay, so what do we have at this point? So the first thing we have is some evidence for genetic clustering at trait-associated loci into schools and friendships. And we also have some evidence that your peers influence your educational attainment. So putting these things together, I think we get some possibility for confounding. So traditionally with GWAS, what we've been interested in is estimating the effect of an individual's own genes on their own traits. But since we also, in this case, at least for certain phenotypes, have some evidence of gene-environment correlation, right? Your gene is associated with the genes of your social alters, and some evidence that the genes of your social alters may matter, right? We have clear evidence, or a clear possibility for bias because we have an omitted variable that we're not including in our GWAS models. So a few kind of notes on that. I think this is something interesting and worth further study. My kind of goal here is not to suggest that the GWAS results are totally wrong or invalid. I just think this is something we now have samples of sufficient size to start to probe a little deeper. I also obviously have talked about things at the aggregate level in terms of polygenic scores, but if they exist at the level of the polygenic score, I think they must exist at the level of individual variance. And so following some variants by small amounts across the entire genome is something that I think would be interesting. So I'm going to stop here. The paper is available in the bioRxiv. This is work I did jointly with several other people: Daniel Belsky, Jason Fletcher, Dalton Conley, Jason Boardman, Kathy Harris. My email address is there. I would be thrilled to get any feedback, comments, or questions you might have. And thank you all for paying attention.
Thank you, Ben.